In the darkness of a cave in Portugal, on a rock wall that has stood undisturbed for thousands of years, there is a small red dot. It is not much to look at. It is a tiny mark, easily missed, a simple pigment perhaps applied by a hand in the distant past. But hidden within that mark, preserved in the stone itself, locked inside a crust of calcite that has formed over millennia, is something that archaeologists have sought for decades, something that seemed impossible, something that defied the assumptions of modern science. It is the DNA of the person who touched that wall, the genetic code, the biological fingerprint of a human being who lived thousands of years ago. And this discovery is about to change everything we thought we understood about prehistoric art, about ancient human behavior, and about the traces that the deep past leaves behind. For centuries, cave paintings have captivated human imagination. Hand stencils, animal figures, abstract marks, pigmented lines that seem to reach across time itself, speaking directly to us across the gulf of thousands of years. Standing before these ancient artworks, people have felt a profound connection, a sense of direct contact with minds separated by an almost incomprehensible span of time. The person who made this mark, the person who blew pigment through a hollow bone to create a handprint, the person who carefully ground ochre and mixed it with animal fat to create a paint. They were here. They thought. They created. They left behind a message for a future they could never have imagined. But despite centuries of study, despite countless expeditions and analyses, science had never been able to directly connect these artworks to the people who created them. We could date the paintings, we could analyze the pigments, we could study the techniques, but we could not identify the artists themselves. We could not know their genetic identity. We could not peer across millennia and look directly into the biological essence of the people who made these marks. Or so we thought. In 2026, an international research team decided to attempt something that had never been successfully done before. They decided to try to extract DNA directly from cave walls. Not from bones, not from sediments, not from tools, but from the rock surface itself, from the very place where ancient human hands had touched stone. The team included researchers from Spain, Portugal, the United Kingdom, China, and Germany. They were part of a larger project called First Art, which focuses on studying some of Europe's oldest cave paintings and understanding the materials and techniques used to create them. The logic was simple. When a human hand touches rock, it leaves something behind. Skin cells, sweat, saliva, traces of biological material. The question was whether these traces could survive for thousands of years, whether they could persist on a rock surface long enough to be recoverable with modern technology, whether the harsh conditions of a cave environment, the infiltration of water, the slow accumulation of mineral deposits, the passage of time itself, would preserve enough genetic material to be analyzed. Most researchers thought it was unlikely. Cave environments are harsh, water flows through caves, sediment accumulates, mineral deposits form, rock surfaces are exposed to countless processes that degrade biological material. Any DNA left on a cave wall would be fragmented, contaminated, destroyed by the passage of time. The odds of finding intact human DNA seemed vanishingly small, but the team decided to try anyway. They assembled a collection of 54 samples from 24 rock art panels in 11 caves across Spain and Portugal. The caves included some of the most famous sites in all of prehistoric art. Altamira, with its polychrome ceiling paintings. Escurial Cave, with its hand stencils and abstract marks. Coveron Cave, with its mysterious figures and symbols. They collected samples from painted surfaces, from unpainted cave walls used as controls, from cave sediments, from bones found in the caves, from a rare ancient airbrush tool, the kind that would have been used to blow pigment onto the rock surface. They applied the most advanced techniques in ancient DNA extraction and sequencing. They used methods developed over decades of paleogenomics research, technology that had been refined through studying the DNA of extinct animals, of Neanderthals, of humans who lived tens of thousands of years ago. They used genetic sequencing so sophisticated that it could detect the signal of a single human DNA molecule amid the noise of contamination and degradation. They searched through the samples with the care of archaeologists searching a site for hidden artifacts. They were looking for one thing: ancient human DNA, preserved on rock, surviving the ages. What they expected to find was perhaps one or two positive samples. Maybe a few results that would suggest human presence, but would be ambiguous, contaminated, inconclusive. Instead, what they found was something extraordinary. 5 samples yielded authentic ancient human DNA. One came from a pigmented calcite crust covering a painted surface in Escurial Cave in Portugal. 2 came from unpainted cave walls in the same cave. 2 more came from unpainted areas near rock art in Coveron Cave in northern Spain. The DNA was ancient. It showed the characteristic patterns of degradation and damage that accumulates over thousands of years. It matched no modern contamination. It was real. It was ancient. It was human. The most striking result came from Escurial Cave, a a small red dot labeled Panel 11, barely notable among the other markings on the cave wall, covered in a crust of calcite, a mineral that forms naturally in caves as water drips and evaporates over millennia. This mineral crust was like nature's preservation medium, like amber encasing an insect. It had protected the biological material inside, isolating it from contamination, sealing it away from the elements, and within that crust, preserved for thousands of years, was the DNA of a human being. What made this discovery even more remarkable was the absence of animal DNA. When researchers sample cave sediment, they expect to find a mixture of biological material, bones from animals that lived in the cave, hair from creatures that sheltered there, traces of the complex ecosystem of life that exists in and around caves. But the sample from the calcite crust at a squirrel cave contained human DNA with virtually no animal DNA present. This meant that the human DNA had not washed in from the surrounding environment. It had not been carried there by dirt on a shoe or a hand. It had come from direct human contact with the rock surface itself. Someone had touched this wall. Someone had left behind their biological trace. And that trace had survived for thousands of years. The researchers themselves were astonished. One of the lead scientists, Itziar Bossoms Mesa, admitted her initial skepticism. "I was very skeptical. I thought, this is too good to be true," she said. She and her colleagues double-checked the results repeatedly, running the analyses again and again, making sure they were not making an error. But the results were clear. Ancient human DNA preserved on a cave wall recovered for the first time in human history. Yet even as this discovery opened new possibilities for understanding prehistoric art, it also raised profound questions. The DNA could have come from the artist who created the painting. It could have come from someone preparing the pigment, grinding the ochre, mixing it with animal fat. It could have come from a family member watching the artwork being created. It could have come from an admirer of the finished work, someone who came to the cave at a later time and touched the wall in reverence or curiosity. It could have come from a later visitor, someone who walked through the cave thousands of years after the artwork was made and accidentally brushed against the wall. The presence of DNA does not automatically tell us who left it. The proximity of DNA to pigment does not prove they were deposited at the same time. But the possibility itself was revolutionary. For the first time, science had a tool that could connect ancient humans directly to ancient artifacts. For the first time, it was theoretically possible to identify the genetic profile of a prehistoric artist, to know their sex, to know their ancestry, to know where their populations came from, to understand who they were as individuals and as groups. This was not merely an archaeological breakthrough. This was a gateway into the intimate lives of prehistoric peoples. The cave at Esquirol held another secret. It had been sealed at the end of the Copper Age, that transitional period between the Stone Age and the Bronze Age. It remained sealed for thousands of years until it was reopened in 1963. This meant that the DNA on its walls could be dated with unusual precision. It was at least 4,000 to 5,000 years old, perhaps older. A genetic message sent across the centuries, preserved by accident, recovered by technology that the ancients could never have imagined. The researchers found something else unexpected. In their searches through the unpainted portions of the caves used as negative controls to detect contamination, they found human DNA more frequently than in the painted sections. 4 of the 5 positive samples came from unpainted walls. This challenges our assumptions about cave art. It suggests that caves were occupied, used and visited by humans in many ways, beyond simply creating artwork. People lived in these caves. They moved through them. They hunted in them. They slept in them. Their bodies shed skin cells. Their hands touched the walls. Their presence accumulated on the rock surfaces in invisible biological traces. The team also found something curious and unexpected: sperm whale DNA. In one of the cave samples, genetic material from a marine mammal appeared. This suggested that whales had been hunted for food by prehistoric peoples, or possibly that whale material had been brought into the cave as part of trade networks. It added another layer to the picture of ancient human life. These were not isolated peoples living in caves away from the world. These were hunters and traders, people with access to marine resources, people with networks of contact and exchange. The implications of this discovery extend far beyond the specific caves studied. If ancient human DNA can be preserved on cave walls, then the walls themselves become an archive written in biological code. Every cave that humans have visited, every cave where hands have touched rock, potentially holds the genetic record of that contact. Thousands of caves across Europe and the world might contain similar traces. Caves that have been undisturbed, protected by their inaccessibility and darkness. Caves that have been sealed by mineral deposits or rockfalls, their contents preserved exactly as they were when humans last walked through them. This opens entirely new possibilities for archaeological research. In the future, archaeologists might be able to enter a cave and extract DNA from multiple locations on the walls. They could build a picture of who visited that cave when they visited, whether they returned repeatedly. They could identify family relationships among people who lived thousands of years apart, connected through the genetic traces they left behind. They could understand movement patterns, migration routes, the flow of human populations across ancient landscapes. They could answer questions about prehistoric demography, talk about the structure of ancient societies, about who was in caves at different times. Preserve DNA as effectively. The conditions have to be right. The environment has to be stable. The rock surface has to be protected. Not every cave wall will yield genetic material. Not every handprint left by an ancient person will survive the ages. There's also the challenge of interpretation. DNA tells us that a person touched a cave wall. It tells us their genetic profile. But it does not tell us what they were doing when they touched the wall. It does not tell us whether they were creating art or simply passing through. It does not tell us their thoughts or feelings or the meaning they attached to the act. Genetic data is profound, but it is limited. It answers some questions, but raises others. Science can tell us who was in the cave. It cannot tell us why. Yet the breakthrough remains extraordinary. For thousands of years, we have looked at prehistoric art and wondered about the people who created it. We have stood before hand stencils and imagined the hand that made them. We have seen pigmented marks and tried to imagine the mind behind them. We have felt a deep human connection across an almost incomprehensible gulf of time. Now, for the first time, we can actually extract the genetic essence of that connection. We can recover the biological code of the person who touched the wall. We can begin to know them not as abstract artists from the distant past, but as actual human beings, with genetic profiles, with ancestry, with place in the human family tree. Completely, but in a way that was impossible just a few years ago. They know that person existed. They know the biological essence of that person. They have recovered a genetic memory of human presence in that cave. As technology continues to advance, these possibilities will expand. Better extraction techniques, more sensitive sequencing, larger databases of ancient DNA. All of these will enable researchers to extract more information from fewer samples, to identify individuals more precisely, to connect ancient people to their archaeological contexts more definitively. The cave walls are archives. The rock surfaces are records. And the DNA preserved on them is a message sent across thousands of years by people who could never have imagined that their touch would be remembered, their biological essence preserved, their presence documented in the genetic code that defines us all. Wall was actual and physical and left behind something that survives even in modern times. The mysterious artists of the deep past are no longer purely mysterious. They are beginning to become real people, individual humans with genetic profiles, with ancestry, with place in our species' vast history. Ancestors. And it reminds us that humanity's story is written not just in stone and pigment and artifact, but in the biological code that connects us all. The deep past and the present are bridged by a molecule so small we cannot see it. And in that molecule, ancient voices speak, ancient hands reach across us,